Welcome everyone. Here we are in chapter 5, so we are now at the end of unit 2. Chapter 5 is the last of our microbiology chapters. We're going to be looking at tissues. So let's start as always with our not really attendance questions. What are the reactants in cellular respiration? And what is the function of microvilli? Go ahead and pause the video and try to answer these two questions. And let's see if we got them correct. What are the reactants in cellular respiration? So reactants, remember, those are what we start with. Those are what is going to go through the chemical reaction. And in cellular respiration, the reactants are glucose, which comes from our diet, and oxygen, which comes from the air that we breathe. Now, I didn't ask, but what would the products be? The products would be carbon dioxide, which is what we're breathing out, and water, which is produced. Now, that's the actual equation for cellular respiration, but that powers all of the mechanisms that make ATP, so you will often see ATP listed with the products. And a lot of times you will see energy to account for that inefficiency. So the energy or heat is a byproduct. Next, what is the function of microvilli? Microvilli, remember, are folding extensions of the plasma membrane, and it is their function to increase the surface area. And a lot of the tissues in this chapter will have microvilli for just that purpose. Now, a lot of this chapter, actually the majority of this chapter, is self-study. We're only going to be actually covering in this lecture a small portion of the chapter, but all of the red material on your outline, the self-study, it is still fair game for the exam. So if you have any questions about any of it, let me know. And a lot of this self-study portion you've actually already covered in lab when you did your tissue lab early in the semester. And you can easily find a lot of this information in chapter 5. For each of the tissues, it shows a diagram or a drawing of the tissue and a photograph of the tissue. But it also shows underneath those drawings the appearance, some example locations, and the functions. So that really will get you through a huge chunk of the self-study part of the chapter. So let's go ahead and begin with chapter 5, tissues, or histology. Histo is a root word that means tissues, and logy is the study of. So histology is just the study of tissues, which is what this chapter is about. Now, tissues we talked about far back in the very first lecture of the semester when we were talking about the levels of structural organization. And we said that tissues were groups of cells with similar shapes, properties, or origins performing a similar function. So tissues are divided really by what do they look like, where do they come from, and what is their function. And there's going to be 4 primary classes of tissues, and that's what we see in our diagram here from your textbook. We have epithelial tissues, connective tissues, nervous tissue, and muscular tissues. And these are really going to be the self-study part of the chapter for the most part. You've already looked at the epithelial tissues, and you've looked at a portion of the connective tissues. Now in lab, you're going to look at some more connective tissues here shortly, and then later in the semester, you're going to look at the nervous tissue and the muscular tissues. But for this chapter, go ahead and learn each of these tissue types and all of their subclasses based on the red part of the outline. But far before we're born, even before we're close to being born, when we are just a little bit more than a mass of cells, we pass through a state where we have this 16-day-old embryonic appearance. And at this point, we're really just 3 layers of cells, kind of arranged in almost a sphere. And at this point, we don't have epithelial tissue, let alone all of the different types of epithelial tissue. We don't have muscular tissue. We don't have nervous tissue or connective tissue. We have 3 very, very generalized tissue types called the ectoderm, which is what we see here in blue, the mesoderm, which we see here in red, and the endoderm, which we see here in yellow. And these are the embryonic tissues. And the embryonic tissues are what all of the cells, all of the tissues in our body came from. And the ectoderm is going to go on to become nervous tissue. And the endoderm is going to go on to make a part of the epithelium along with both the ectoderm and mesoderm. The mesoderm by itself is going to go on to become muscular tissue and connective tissue. So this early on in our development is basically what all of us looked like. And all of these tissues that are in our body now can be traced back to one or a combination of these 3 tissue layers. So first, let's talk about epithelial tissue. And again, you've already seen these in lab. And epithelial tissue comes in 2 forms: covering epithelia, which is on the surface or the exposed portions of our body, and also lining hollow body cavities. So, for example, the very surface of our skin, or lining the inside of our mouth, or the inside of our digestive tract, the inside of our respiratory tract, the inside of our blood vessels, or covering our blood vessels, covering most of our organs. So these are epithelial tissue locations. This is where we find covering epithelia. But we also have glandular epithelia. And glandular epithelia is what all of our glands are made from. Now, the functions of epithelial tissue, there's quite a few. Epithelium functions as protection. So since it's, kind of wrapping or lining all of these different places in the body, it helps resist things like abrasion, friction, stuff like that. When we look at skin later in the semester, we're going to see how the skin is shaped or it's built with a certain type of epithelium that can withstand a lot of abrasion. Also, secretion. So glands, which are epithelial tissues, will secrete products that they've made. Excretion. Excretion is similar to secretion, but in secretion, it's something that the body is going to be using, and excretion, it's something that the body is getting rid of. Absorption. As food is digested and it moves through our digestive tract, then the nutrients from that food are going to be absorbed through epithelial tissue, ultimately into our bloodstream. Filtration. Our blood, for example, is cleansed by our kidneys, and it's going to be cleansed by special structures in our kidneys made of epithelial tissue, and that process is filtration. And sensation. Most of our epithelial tissues have a lot of nerve endings. In our skin, for example, there's a lot of nerve endings that can detect things like pressure, heat, cold, things like that. So sensation. Now, it's not the epithelial tissue itself It's the nerve endings that are in the epithelial tissue. But since epithelial tissue has a lot of nerve endings, we attribute some sensation to that epithelial tissue. So let's look at some characteristics of epithelial tissue. It has polarity. There's that word again. I told you we would see polarity or polar a lot this semester, and it's always going to mean the same thing. What did polarity mean? It means different regions of something have different properties. And that's the case with covering epithelial tissue. When epithelial tissue, covering epithelial tissue, is looked at, it has both an exposed surface, which is either what's exposed to the environment or to a body cavity. And then it has another side that is attached to whatever is deep to it. So for example, over here is a drawing of some epithelial tissue. These pink cells look like probably simple columnar epithelial tissue. And then above that is nothing. So maybe this is the inside of your digestive tract. If this were a different type of epithelial tissue, maybe up here would be the outside of the body, or maybe it's the inside of a blood vessel. But it's something that is exposed. But if we go deep, well, down here is connective tissue. So this is whatever that epithelial tissue is attached to. Now, we can see up here, if this area is exposed, and down here, this area is attached to other stuff in the body, well, these two surfaces are different. We would say this epithelial tissue has polarity. This side of it has some different properties than this side. So let's give some names to what I keep saying, this side and that side. The side that is exposed either to the environment or to a body cavity is called the apical surface. Apex usually means top, and in this drawing it is the top of the drawing. This is the apical surface. The other side, the side that is attached to whatever is beneath it or deep to it, is called the basal surface. So we have an apical surface and a basal surface when we look at covering epithelia. And they have different properties. Apical surface, since it's exposed to the environment or a body cavity, that will be where we will find things like microvilli or cilia. Now, not always, but it's really common to find microvilli or cilia along these apical surfaces. Now the basal surface, since that's facing other parts of the body, since this is attached to whatever is deep to it, down here we find some structures that help with anchoring these cells in place. So one thing that we will find is the basal surface will secrete this substance that's kind of like a jelly almost, called the basal lamina. The basal surface secretes the basal lamina. And the basal lamina has a lot of proteins like collagen. It has a lot of glycoproteins. So we have kind of this sticky jelly-like substance here. Now if we look at the yellow and blue area of this drawing, this is going to be connective tissue. Almost always, epithelial tissue will have connective tissue just deep to it. And almost always, that connective tissue is areolar connective tissue. Not always, but the vast majority of the time, areolar connective tissue is just deep to whatever epithelial tissue we have. But regardless of what connective tissue it is, the connective tissue will also secrete this sticky substance, very similar to the basal lamina, but this is called the reticular lamina. Still lots of collagen, lots of glycoproteins. And the basal lamina and the reticular lamina will grab hold of each other like Velcro. And what that does is it allows the epithelial tissue to be anchored to whatever is deep to it. The basal lamina and the reticular lamina act like Velcro. And these two lamina together make up what we call the basement membrane. So the basement membrane helps to hold the epithelial tissue in place. But in addition to that, the basement membrane acts as a barrier. Since this epithelial tissue is exposed either to the environment or various cavities of the body, there could be things that might cross the epithelial layer. And we don't want it to be able to get to the rest of the body. So the basement membrane acts as a barrier, as a wall, to prevent things from crossing. Now, one thing that comes about in epithelial tissue really, really often is epithelial tissue, one, where it is exposed to the environment, and two, because it's subjected to damage, it's going through mitosis quite a bit. We will see a lot of cancers of the epithelial tissue. Epithelial tissue is really prone to cancer. And the basement membrane is really good at keeping those cancerous cells confined to the epithelium so that it cannot cross and get into either the blood supply or the lymph system where it can easily spread to the rest of the body. But some cancers actually develop the ability to move. And if they develop the ability to move, sometimes they can develop the ability to cross the basement membrane. And when they do that, we say they are metastatic. Metastatic is a cancer that can move from one place to another. And sometimes epithelial tissues will develop a metastatic cancer that can cross the basement membrane and then migrate to other places in the body. So one example of this is a skin cancer called melanoma. Melanoma is highly metastatic. It is really good at crossing the basement membrane, getting into our lymph flow, and moving elsewhere in the body. Now, continuing with some of the properties of epithelial tissue is avascular. That means it does not have a blood supply. A lot of times when we see a at the beginning of a word, it means without. And vascule means blood vessels. So avascular is without blood vessels. Now, there are blood vessels nearby. They're just not going to actually be within the epithelial tissue itself. Epithelial tissue is innervated. That means it has nerve endings. And as we mentioned earlier, it tends to have a lot of nerve endings. And epithelial tissue is highly regenerative. We will talk about regeneration towards the end of this lecture, but regenerative just means if this tissue is damaged, it can be pretty easily replaced with the exact same tissue. And as you saw in lab, epithelial tissue is going to have some easy ways to classify it. And one of those ways is by how many layers are there. So if it's a single layer, we call it simple. If it's multiple layers, we called it stratified. If it was a single layer that looked like multiple layers, we called it pseudo-stratified. And that's what we see here in the middle. This at first glance looks like multiple layers. We have some cells up here and we have some cells down here. But how do you know if it's simple or stratified? How do we know that this pseudo-stratified is really only a single layer? Look at the basement membrane. Every single one of these cells is attached to the basement membrane. So this is really only one layer. Over here in the stratified, this row here at the bottom is attached to the basement membrane, but these others are not. So there are multiple layers. And then we also classify by cell shape. We saw squamous or squamous. I say squamous because it sounds like squashed, which gives me an idea that these cells look very, very flat or squashed. Cuboidal, which means that the cells are roughly the same height as they are wide. And columnar, meaning the cells are tall and thin like a column. So those are how we can classify epithelial tissues. Now, the actual epithelial tissues themselves, we are not going to discuss in lecture. You can learn those on your own in self-study. So now changing to the next tissue type, we're going to talk about connective tissues. And connective tissues have some characteristics of their own. When we were looking at epithelial tissues, they were highly cellular. Let's go back to look at this picture here. Everything we see here is a cell. Lots of cells packed really closely together. But when we look at connective tissue, we say that it has very low cellularity. When we look at the tissues under a microscope, the cells are very spread out and there's a lot of open space in between them. And that open space is what we call the extracellular matrix. We talked about that back in chapter 3. The extracellular matrix, remember, was the area outside of and between the cells. The extracellular matrix is really extensive in connective tissue. So lots of extracellular matrix, just a few cells scattered here and there. There's a wide range of vascularity. Some connective tissue has a pretty good blood flow, lots of blood vessels. Some connective tissues don't have any blood vessels at all, such as cartilage. If you've ever damaged or known anyone who has damaged a cartilage in your knee, you know that it is really slow to heal because it doesn't have a good blood supply to bring in all the nutrients needed for that healing process. And connective tissue has a common origin, even though they all look really, really different, as we see here in these pictures here. They all come from the mesoderm in that embryonic layer. So things like bone versus fat versus tendons versus blood, they all look really, really different and they don't seem like they have much in common. But they all have that common origin in the mesoderm. Connective tissue has a lot of functions, more than pretty much any other tissue. For example, first, it binds organs. So it helps surround organs and kind of give them shape and hold them together. Remember, it's just underneath the epithelial tissue. It also holds organs in place. Organs in our abdominal cavity, for example, are held in place by some connective tissue. Support, similar to what we were just talking about with the binding organs, the support helps stabilize, helps keep everything in place and in the right orientation. In our knee, for example, there's a lot of connective tissue surrounding our knee to give it support. Physical protection. Things like our bones, our rib cage protects things like our lungs and our heart. Immune protection. In our blood, we have special structures called white blood cells or leukocytes. And blood is a connective tissue. And these leukocytes, these white blood cells, are our immune system. Allows for movement. Now, the connective tissue doesn't actually move. Any movement that happens in our body is the result of muscles, but the connective tissue, things like our bones and our ligaments and tendons, they all work together with our muscular system to allow for movement. When our muscles contract, it helps move the angles of our joints. It moves our bones. Storage. This is specifically talking about bones and fat. The bones store a lot of minerals, and the fat stores a lot of energy. So we can kind of think of our connective tissue as a bank. When we have too much of something, we can store it in either our bones or our adipose tissue. When we need some nutrients, we can withdraw it from our bones or our adipose tissue. Heat production. This is also called the calorogenic effect or the thermogenic effect. Heat production is a big job of a tissue that a lot of people don't know about called brown fat or brown adipose tissue. And brown adipose tissue or brown fat isn't like the fat that we tend to think of, which is called yellow fat. Brown fat is actually really, really metabolically active, and it creates a lot of heat. Transport. Well, our blood carries everything around our body from place to place, kind of like a highway system. And blood is a connective tissue. So our blood is the transport system of our body. Now, we're not going to talk about the individual types of connective tissues. Again, that's self-study. Nervous tissue and muscular tissue, also self-study. We will be looking at those in their own chapters, though, coming up just past the halfway point in the semester. So let's talk about cell junctions, and really we're going to be talking about epithelial tissue here when we talk about cell junctions. Now, we will see these cell junctions in muscular tissue, in nerves, but primarily we're going to be talking about epithelial tissue. So when cells are joined to each other, they can be joined in different ways. And we see some examples here. First, we'll talk about tight junctions. And tight junctions are just what they sound like. The cells are joined to each other very, very tightly and very, very closely. So we can see that right here. These two membranes are so tight, it almost just looks like one membrane. And we find places, or we find tight junctions in places where we do not want something to be able to get between the cells. For example, in the intestines in our digestive tract. We do not want those digestive enzymes and acid to be able to leak between the cells because it could get out into our abdominal cavity and do a lot of damage. So we have tight junctions there to prevent that. Next, desmosomes, or sometimes you'll hear them called anchoring junctions. Desmosomes, we can see there is some space in between the cells. But desmosomes, they're still going to hold on to each other very, very strongly. But there's a little bit of a difference with tight junctions and desmosomes. In desmosomes, there's a lot more freedom of movement. The cells, while they're held to each other very, very strongly, the two cells can move about kind of independently of each other. For example, I like to say, think of desmosomes as if you've ever been on a boat. At the end of the day, when you're ready to get off the boat, what do? You go up to the dock, you throw a rope over to the dock, and you tie it around that little hook. Well, the boat is going to be very snug. It's going to be, not floating away, but the waves will still move that boat about. It's not going to float away, but it's going to be held in place even though it can move some. That's how desmosomes work. And we find desmosomes, a perfect example is in the heart. Heart muscle cells join end to end. And there's a special structure there called an intercalated disc. And at the intercalated discs, we find desmosomes. When heart muscle contracts, they pull on each other, and we don't want them to snap apart. Desmosomes are really, really strong. They will keep those heart muscles from snapping apart. But it gives the heart muscle some degree of movement so that those cells can move around a little bit. Now, we also will see something called a hemidesmosome. A hemidesmosome is basically half of a desmosome. And instead of being joined to a neighboring cell, we will see that it's joined to something like the basement membrane. So it's going to keep this cell anchored to its surroundings. And finally, gap junctions. Gap junctions don't really serve to hold cells together. Instead, their function is to allow stuff from one cell to pass through a tunnel into the next cell. Now, a place that we find gap junctions is also in the intercalated disc where two heart muscle cells join together. We'll also find these in certain neuron junctions, so nerve cell junctions. And here, this makes it really, really efficient for things like ions, charged particles, to move from one cell into the next cell, carrying that positive or negative charge with it. So basically, that means that electrical activity in one cell can really efficiently spread into the next cell. And as an electrical charge spreads through your heart, these gap junctions in the intercalated disc make it happen really, really easily. Now let's talk about tissue growth. Tissue obviously has to grow either because you're getting bigger or because you're replacing damaged tissue. But there are different types of tissue growth. First, there's something called hyperplasia. And in hyperplasia, we are having an increase in the number of cells. Instead of having 2 cells, they're going to divide to make 4. And what we're doing is increasing the tissue by increasing the number of cells that make that tissue. Next, there is hypertrophy. In hypertrophy, the number of cells stays the same, but the cells themselves get bigger. So here we still have, 10 cells and 10 cells, but the size of the cells are bigger. We have neoplasia, which we see down here at the bottom. Neoplasia is really tumor development in a cancer. Neoplasia is tumor development, and a couple of things happen there. First, the number of cells increases, but when cells divide, remember when we were talking about mitosis, cell division, we said that most of the time cells in your body are not dividing? Well, here we have a lot of cells that are dividing to produce more cells that are still dividing. So we have an increase in the number of dividing cells. And even though we're having an increase in the number of dividing cells, the result is more dividing cells and a really, really disorganized product. Here, what it's trying to show you is the green is one type of cell and the pink is another type of cell. And you can see that the types of cells are kind of in their own region. Over here, the pink and the green are all just kind of mixed together. So the cell types aren't following any sort of organization. It's very disorganized. And that's common in cancers. Let's talk now about tissue development. So we saw that early on, we were just a group of 3 types of tissues. Before that, we were actually no real tissue type. We were just a lump of cells. So we go from no tissue types to 3 tissue types to many tissue types. And that requires the tissue to develop. And one thing that we will see is differentiation. And differentiation is what happens when we go from a tissue type that is unspecialized, so either that original lump of cells or those 3 tissue layers, to a very specialized type of tissue. So when we went from one of those layers to a connective tissue, that was differentiation. Another type is metaplasia. Metaplasia is when we change from one specialized type of tissue to another. So here is that lump of cells that I was talking about before we get to that 3-layer embryonic tissue layout. This is called the morula, and this is very, very early on in embryonic development. Here we have, it says about 16 to 32 cells, and these cells are not specialized, they're not differentiated at all. And they have what we call developmental plasticity. Developmental plasticity just means they can become a number of different specialized cell types. For example, this cell could go on to become anything. It could become any cell in your body. It doesn't matter what cell it is. This cell could potentially become any one of those cells. Now, once we get to that 3-layer structure that we saw, we still have plasticity. It's just a little bit more limited. We saw that each of those layers could become a certain tissue type, but they're limited in what tissue types they could become. As we age, we get fewer and fewer and fewer stem cells. And not only are there going to be fewer stem cells, they have much more limited plasticity. So let's look first at embryonic stem cells. And let's look at this morula, this completely unspecialized, this completely open as far as what path it could take group of cells. We say these cells are totipotent. Totipotent means that these cells could become any cell type, any tissue type, any type of cell or tissue in your body. As they go and have a little bit more differentiation, and eventually they get to that 3-layer structure, then we say that the cells are pluripotent. Pluripotent means that it can become any type of embryonic tissue. So we are now more specialized. We can still become any type of embryonic tissue, but it cannot become any tissue type. Once we are, more resembling a human, and then after birth and we become even more and more and more specialized, we still have some stem cells in our body. We call them adult stem cells. And throughout this semester and next semester, we will see quite a bit of those stem cells. But adult stem cells are really, really limited. Adult stem cells can only become a limited type of cells. For example, we have multipotent stem cells. These are stem cells that when they divide, they can produce several types of cell, but not any type of cell. For example, in our next chapter, in the skin, we have a layer of stem cells just at the bottom of our epidermis. And those cells divide, but they can only produce the different types of stem cells. They cannot produce any other cell type. And finally, there are unipotent stem cells. And unipotent stem cells can only produce one single cell type. For example, in males, the testes have cells that are dividing to produce sperm cells. But that's all that they can produce are more sperm cells. So they are unipotent. So what we can see is once we get past this stage, we are more and more and more limited. The earlier on, the broader range of cell types that each cell can produce is. But the older we get, and when I'm saying older, I don't mean elderly, I mean days old, we are becoming more and more and more limited. So let's use that as a thinking cap question. Why are stem cells such a goal for nerve damage, such as in spinal cord injuries? And you've probably heard about debate, is it ethical, should it be done? And we're not going to talk about that. We're not going to talk about the ethics or should it be done. We're going to talk about for the people, the scientists that want to be able to use stem cells. Why is it so important for them to use stem cells? Why is that a goal for them? So if someone has a spinal cord injury, why do scientists want to study stem cells for that purpose? So go ahead and try to come up with an answer for this before listening to the answer. Well, first we have to think about nerves, which primarily is what the spinal cord is, or this would also apply to the brain. If, let's say, someone severs their spinal cord and they become paralyzed from the waist down. If they severed their spinal cord, then they are going to be paralyzed from the waist down for the rest of their life. Nerves, when they are destroyed, they do not regenerate. They are dead and they will remain dead. Now, when we talk about nerves later in the semester, we will see that if there's nerve damage, well, sometimes we can recover from that, depending on what type of damage it is. But nerve death, if we completely destroy neurons, then they're gone. Neurons do not go through mitosis. Neurons cannot be replaced. So, how does that tie into stem cells? Well, if we have this, if we have morula, or if we have these embryonic layers, either of those cell types could potentially become nerve cells. They are not limited in what they can become the way neurons are. Neurons will not divide to produce new neurons. But morula cells or embryonic stem cells can divide to produce nerves. Well, how does that work? The way embryonic stem cells work are if you place some of those cells into a tissue, then it basically looks around and says, oh, I'm surrounded by nerves. I guess I'm supposed to become neurons. And it starts to divide to produce neurons. Adult cells cannot do that. Even child cells cannot do that. If there is nerve damage, that nerve damage is forever. But embryonic stem cells, if you put those in, they will start dividing and producing neurons. So that is why scientists have this goal of working with embryonic stem cells, because embryonic stem cells, there is unlimited possibility with what kind of damage we can fix. Tissue repair. So now moving on to, we have tissue that gets damaged. How do we fix that damage? Well, the tissue is going to start to fix itself, and it depends on a couple of things. One, it depends on what type of tissue is damaged. And two, it depends on how severe the damage was. And when that tissue starts to fix itself, then we're going to go through two different things. We're going to get something called regeneration. We mentioned this earlier. Regeneration is replacing the damaged tissue with the exact same tissue. For example, if you damage stratified squamous epithelium, you replace it with more stratified squamous epithelium. That's regeneration. Fibrosis is when you replace the damaged tissue with fibrous connective tissue. For example, if you damage your stratified squamous epithelium and you replace it instead with some sort of fibrous connective tissue. But in reality, it's not one or the other. There's always some regeneration and some fibrosis. But depending on which tissue type and how severe, maybe you'll get more regeneration, or maybe you'll get more fibrosis. And we can see the 4 steps here. So what happens? Well, first, when you get damage, there's going to be some bleeding. Now, maybe that bleeding spills out onto the surface of your skin, or maybe it's internal. It doesn't matter. When there's damage, there's going to be some bleeding. And as that bleeding fills out, it's going to kind of fill in the gaps and start to form a scab. And in addition to stopping the bleeding, the scab is also going to be full of macrophages, cells that are going to perform phagocytosis. Phagocytosis to get rid of bacteria that may have invaded the cut, or to get rid of damaged cells and tissue. They're just there to clean up. Then we're going to have formation of granulation. So that's just growing new blood vessels and new fibers. And we're going to start to produce something called fibroblasts. Well, anytime we see blast, B-L-A-S-T, at the end of a word, it means to give rise to give birth to, or to form. So fibroblasts are cells that form fibers. These cells are going to start laying down things like collagen fibers to help patch up this damage. And finally, regeneration and fibrosis. So this is when we start to have a lot of mitosis occurring, either regeneration to replace the tissue with the same type of tissue, or fibrosis to where more and more and more fibers are going to be laid down. And again, it depends on the tissue type and the severity of the damage. In the end, you get something like a scar, possibly. So what's a scar? A scar is what happens when there is more fibrosis. If you've ever cut your arm and a few days later, all of a sudden you can't even see where that cut was. Well, that's a lot of regeneration and hardly any fibrosis. But if you cut your arm pretty well, then, a few weeks, even a few months, or a few years later, you may have like a rough, jagged spot there. That's a scar. And that's what happened when there was very little regeneration and a lot of fibrosis. So a couple of more thinking cap questions. Why do scars feel different from the surrounding skin? And why is scar tissue on the heart such a problem? Go ahead and try to answer those. The first one we just talked about. So why do scars feel different from the surrounding skin? Well, because it's a different tissue type. Normally, the outer layer of the skin, the part that you feel, is stratified squamous epithelium. But if you have a scar, well, now we have stratified squamous epithelium with a lot of fibrous connective tissue built into it. And next, why is scar tissue on the heart such a problem? Well, the heart is almost entirely made of cardiac muscle. And cardiac muscle is one of three types of muscle. It's there to contract. It's there to cause movement. But scar tissue results when there is death of cardiac. Cardiac muscle is not at all regenerative. When cardiac muscle cells die, there is no regeneration. It is all fibrosis. And if it's all fibrosis, then when that part of the heart dies, such as after a heart attack, we don't replace it with more cardiac. We replace it with fibrous connective tissue. And fibrous connective tissue does not have the same properties or functions as cardiac muscle. When a part of the heart dies, that part of the heart is not going to be able to contract and relax anymore. So that's why it's such a problem. Now let's talk about death or damage to tissues. And there's a lot of different types of damage and different ways that tissue can die. These are just a few. So first, let's talk about something called atrophy. Atrophy is just shrinkage of tissue. And maybe that shrinkage is due to a decrease in the size of the cells or a decrease in the number of cells. Either thing is going to result in a decrease in the tissue size. And there's a couple of different types of atrophy. There's something called senile atrophy, and that's just something that naturally happens with age. As you age, your tissues are going to shrink just from the normal aging process. But there's also disuse atrophy, and this is more commonly just referred to as atrophy. If you've ever broken an arm and you couldn't move it for a long time as it healed, well, once that cast is off, you'll see that the muscles in that area have really, really shrunken. It looks kind of withered. Well, disuse atrophy is from lack of use. This also happens in people who are bedridden, people who are paralyzed. If they can't move their muscles, then those muscles are going to shrink. Well, that's tissue damage or tissue shrinkage. Let's talk about tissue death. Now, let's talk about first something called necrosis. Necrosis is premature pathological death of tissues. And there's a few types of necrosis. There's something called infarction. Infarction happens when there is sudden tissue death, and that tissue death is because of a loss of blood supply. The actual name for a heart attack is myocardial infarction. Myo means muscle, cardia means heart. So this is death of heart muscle due to loss of blood supply. If a blood vessel is completely blocked, then blood is not going to be able to bring oxygen and nutrients to that muscle. If you don't have oxygen, what happens? Well, as we saw in the previous chapter, we don't get ATP. And we start to get lactic acid building up. So that heart muscle is going to die. So that is infarction. We could also have something called gangrene, and that's what we see over here in this nice picture. Gangrene is from infection that comes about because of a disruption in blood supply. Now, disruption could mean that it is completely blocked off or that it is just really, really diminished, but then infection sets in. And there's several different types of gangrene. This is called dry gangrene. There's something else called gaseous gangrene, in which swelling, even, and this is really gross, but we can get actual small explosions. You can see pictures, if you're interested in that, in people's legs. Sometimes they'll become gangrenous and pressure builds up inside the leg and it starts to swell, and ultimately it will rupture. And then there's also something called decubitus ulcers, more commonly just called bed sores or pressure sores. And this is a form of gangrene if someone is bedridden or stationary for a really long time. That pressure in one spot is going to cause the gangrene. Now, Ideally, if someone is bedridden, they should be moved regularly to keep the pressure from being in one spot for too long. But unfortunately, this is something that's really common in nursing homes. If the patients are not moved enough, they will get these bed sores. All right, well, that is the end of Unit 2. So we are now ready for Exam 2. And then after Exam 2, we start the second half of the semester. We will start actual systems biology. So if you have any questions at all, please let me know. The exam is going to cover everything since the last exam in depth. So all of the stuff that we've covered since Exam 1 is going to be the focus. But every exam has a little bit of cumulative material. So there's still going to be some material that was also on Exam 1. It's not going to be any certain focus on older material. It could be anything from older material. But the focus is going to be on everything that we've covered since Exam 1. Okay, so if you have any questions at all, let me know. Otherwise, I will talk to you next time. Take care.